Well, today we're going to do the toggle, which we didn't get to last week. And I think it's important to go over some of the basic concepts. So for some people, this may be a review. For other people, it's a chance to practice a little bit. As always, we're recording these and I'm trying to get them edited and put them up online for you. I'd like people to give me quick updates on their projects. I've added a little tiny exercise which might be helpful when people are preparing notebooks to use for projects and also for homework. And one thing that would be helpful to me is if you're doing your notebook in Colab, share the link to the notebook. And so again, today we're going to do the things that we had talked about doing last week, which is talking about the toggle, starting out with some basic exercises for the toggle, some of which we already had worked on a little bit. I'm going to emphasize thinking about the toggle circuit a little bit, how to interpret the mathematics. And then we're going to do some exercises on phase portraits, and I'm going to give you the code for that. I'm not going to assume that you're going to develop that on your own. If you want, there are a whole bunch of slides in this slide deck that in the back that go on to how to do it. And then I would like to get to the effect of noise. And as I say, I didn't write the homework up in advance because I didn't know how far we'd get. And so I'd rather decide on the homework after we see where we get to. So maybe we'll start out by just asking people for quick updates. I've talked to two of you individually.
I think I had this like grand idea and didn't really understand the basics. So after talking with you last week, I think I've settled on like really wrapping my head around kind of FBA and just how people do a lot of this stuff. So I was thinking of doing a project testing the effect of the different like constraints you can add to FBA and how that can either like make the model better or worse. So using a single model of E. coli, a genome scale metabolic model, I was gonna do the FBA analysis, enzyme constraint FBA analysis, and then a thermodynamic FBA essentially. It's just like different essentially constraints you can do on them. And then combine the last two and see how doing both of them work. So doing that under like different media conditions, you can see like how the whole network works under all and like what changes when you add those constraints. And so there's a couple papers I found where people have done something similar. So I have a little bit of a like backbone to kind of go off of. But yeah, and then as far as the stuff this week of like the parameter things, there's different parameters in each one, and I'm not entirely clear on what you can do like scans like for the thermodynamics on the ranges of metabolites, which let the general like free I don't know the direction of the reaction or something like that. The enzyme constraint one, you can change the values of the K-cats and the like total enzyme and stuff like that. So there's a couple different things you can play with to kind of do scans and see, and I think tie it back to like E. coli's very well studied. So there's a lot of like growth data and stuff like that. Should be able to link out.
Again, I recommend that. I think that sounds exciting. I always recommend starting with a paper where you can actually grab. Ideally, they'll have code, but if they don't have code, at least they have algorithms, so you can actually replicate a result from a single paper to start with. And there's a technical issue, which is there's a difference between, and people unfortunately use the terms inconsistently. There are two terms in science, one of which is reproducibility and one of which is replicability. And one of them means to redo something with the same methodology that the original was done in. So, for example, if you can download the code that some people have published for a computer simulation and run their code, that would be replicating their results if you got the same results. If you took their algorithms, but you wrote your own code, suppose they hard-coded their model at MATLAB, but you put it into Antimony, for example. And so you solved the same concepts with a different methodology, that would be reproducing their results. And of course, the ability to reproduce something tells you something about a little bit more about the fundamental science of it. Because if you're replicating it, if there's an error in their code, you'll get the same error. And so the fact that you get the same results that they got doesn't tell you that the results are necessarily correct. It does tell you that technically their code works. Now, there is the unfortunate detail that some people use the terms reproduce and replicate exactly the opposite way. And so when you see somebody talk about reproduction or replication, you have to look at which term they're using to mean which concept. They're always different, but about half of the people use the terms one way and half the people use them the other way. So I mention this because there's this, I think I sent it around to you, this cover story in the SIAM newsletter on how to publish and reproduce models. And that I thought was a nice one-page introduction to some of the issues that you face. And I encourage you to read that. And it may help you also when you're reading other people's papers. For example, when we sat down with Varalakshmi the other day to talk about some of the papers that she was looking at for her project, some of those papers were actually pretty hard to interpret. And so one would like to try to avoid having that happen. And I'm not saying that it's easy to publish things in a way that is intelligible and reproducible. But it is actually of importance. People may have heard of the term crisis of reproducibility in biology. There's a lot of concern that many, especially medical experiments, when people try to reproduce those experiments, they don't get the same results. Now, with computation, you would think perhaps that it should be easy. Since it's code, the code should do the same thing. But in fact, very often, as we found out even in this class, if we wanted to run Tellurium in Colab now, we had to reload some of the packages in a different way because there were package incompatibilities. Compilers change, processors change. Sometimes the code that people publish isn't quite complete or isn't correct. And so even with computational models, it's not always possible to replicate results in a simple way. And so that website we were working with yesterday, the Biomodels website, is designed, and that's the purpose of this SBML exchange language. It's to write model descriptions that are portable, and they don't specify the numerical way that those models are solved. And so if you download an SBML model and run it, you're actually doing a reproduction of the simulation rather than replication. You could run it with a different solver, for example. And so that's something to think about a little bit. I think that that's fine. I think starting with reproducing or replicating the results in a single paper and really understanding that already is not trivial. And if you've got that under your belt and then you want to expand your work, that's fine. But I think you're right that focusing on a single paper to begin with is a good idea to make sure that you're solid with that. Especially if this material is unfamiliar to you, then there's a victory in understanding one paper really well. Okay, well, that sounds great. Let's make an appointment and go over it in a little more detail together. Pedro may have some ideas about that too. That sounds fine. Okay, so today I want to remind you, because we're going to be using these ideas that we've been working on now for some time, about this idea of rate equations for gene regulatory networks. And we're going to be doing this translation. You already did it in the homework to some extent, which is you have this concept that a gene 1 regulates gene 2 either as an activator or an inhibitor. And that's interpreted as nothing goes to gene 2 because you don't model the species that are used up in the production of the protein. And then fundamentally, P1 will come in with a rate law, and those rate laws typically for gene regulation are Hill functions. And in this class, we're typically going to use these symmetrical Hill functions where we have P1 to the n over K to the n plus P1 to the n. You'll find in a lot of the experimental literature, they will use hyperbolic tangent instead of these power law forms. They have basically the same shape, and they behave in a very similar way. You can certainly implement a hyperbolic tangent in antimony. It doesn't take any more effort. But to me, the P to the n over K to the n form makes it clearer what's happening. The K to the n is the half maximum concentration. The n then is the steepness of the slope. And so the form is similar and simpler between the activator and the inhibitor. And so we're not going to use the tanh forms in class very much, but people should be aware that those exist too, and they behave very similarly to the forms that we're using here. And again, this is the form that you get from those Hill functions. This is for a Hill exponent of 8. And I'm comparing those to a Boolean switch, where for the activating Hill function, it turns on at K1. For activating Hill function, inhibitory Hill function turns off at K1. And then we looked at biomodels, and we had problems because biomodels didn't seem to want to load very fast last week. In the news today, biomodels is moving from England to Florida. It's going to be partly managed by one of my old students, actually. The person who was its original developer, Rahman Sharif, is going to continue to be the manager of it. And then we also talked about how to convert SBML to antimony and back. Tellurium allows you to load SBML models directly. The problem with SBML models is they're not very easy to read. And so this r.get antimony converts SBML to antimony, and that makes it a little bit more human readable. And similarly, you can go the other way. There's an r.get SBML that'll convert the other direction. And we also used this nice online web tool that lets you convert back and forth. That was written by Bart, who's somebody we've worked with over the years. And actually, that seemed to work pretty well when we tried it last week. We also talked a little bit about time-dependent expressions in antimony, the colon equals notation. You can also define functions. So there's you can define functional relationship functions as well, which are not the same as a colon equals. A function definition is like a function definition in C++, for example, or Python. It doesn't explicitly have a time dependence associated with it. And we also talked about the fact that you can write differential equations, first order time derivatives directly with the apostrophe as a notation. So something that's not antimony, but I think is worth trying out here, and I added this at the last minute, so we'll see whether it works for people or not. And people will have to fire up their Colab to do this, is that I noticed that for drawing diagrams inside of Colab, you can certainly take images, screenshots of the network, and put them into Colab cells. That's possible. And of course, that's something people do a lot. But there is a package called GraphViz that allows you to draw simple network diagrams. And I wanted everybody to try this out. And I'm certainly not an expert in GraphViz. And so I'm not suggesting that I'd have to learn with you for all the fancy things. But for simple things that we can do it. And so I'd like people to try this. Import GraphViz, and then there are a number of possible ways to do the call for the display. I would suggest from ipython.display, import display will work. Then you put the description of the network that you want to plot into a string, so it's like antimony in that sense. And then you apply the GraphViz source to that string, and then you do a display. So why don't people try that and see if you can generate the diagrams that we had for the homework from 2 weeks ago. A would be pretty straightforward. I think it would be A goes to A, and the arrowhead would be of a T-type. B would be A goes to A, and the arrowhead would be a dot, and then see if you can get those to work. And if you get through all of that before your classmates, I would suggest one thing: you can look online at the software, at the documentation software for GraphViz, and there are options for how to lay things out and change line widths and change colors and all sorts of other things. But this way, if you want to put a network plot into your Colab notebook and you don't want to have to cut and paste it from somewhere else, you might want to change the network at some point. It allows you to do it pretty straightforwardly. So when I do it, it works. And if I wanted to do one of the other ones, let's see if I can do just A goes to A. It works. Pedro, do you have a favorite graph plot, network plotting tool?
So either GraphViz, because you can, especially for very large networks, it organizes for you automatically, which is very, very good. And you can also use ChatGPT to generate it. So you can, for example, give ChatGPT your Tellurium code and it generates a GraphViz diagram. There is a limitation, which is you cannot point arrows to arrows, which is something we usually need, but you can use junctions. So you create like nodes and then you combine things like this. So you can use like dummy nodes where you receive different signals. And if you want to do something very beautiful and customized, then I use biorender. And probably one could also get a screenshot of equations from a paper, then use MathPix to convert to a LaTeX text. And then from LaTeX, you paste on a ChatGPT prompt and ask to convert that to a diagram in GraphViz, and then that will automatically plot the diagram. MathPix, yeah, MathPix.
The website.
No, it's an app. You download the app. Let me show you.
You want a screen share?
Oh yeah, yeah, that's the better way. Wait a second.
You were able to generate all of these diagrams? Well, that's what I said. If you've gotten through the first one or two, then you can do the other ones. They don't take. The nice thing about GraphViz is the notation is like Antimony. It's very simple.
This is MathPix, and let me.
Here, I'll put
Yeah,
it
this is how it looks like. They have this snip from screenshot
up.
function up here.
Wait a
You should
second.
be able to see from the screen share, or you can also watch from a computer. But are you guys in the Zoom meeting?
There you go. Go ahead.
Okay. Yeah, you can look there, or you can look here too, either one. So let me try to do this. Let me go to a paper. Let me perhaps, I don't know, I'm gonna get a random paper, cancer model ODE. Let's see. Let's do P53 ODE network.
Progress.
Let's see if I can find something easy to. Easy to capture.
There you go.
Right here. So I can we increase this? Maybe not. I will take a screenshot of this. And then I will paste. Oh, sorry, I have to use that. Okay. That's the function to take the screenshot. So now I take a screenshot of this. And it gives me more or less what's going on there. This is already very helpful, but let's just do a
So it converts the image to LaTeX, huh?
yes. And it also exports to Word. So if you're using Google Docs, for example, you click Open Docs or copy MS Word, and here are the equations in Word. Yeah,
And what is the utility called?
MathPix.
MathPix.
Yeah.
PIX. MathPix. MathPix snipping tool. Yeah. So for some reason this is cutting there. I don't know why, but okay. So now I'm gonna copy this and I will ask ChatGPT or Copilot to generate a dot out of that. So let's select ChatGPT here and convert this ODE system to a graph this diagram. and I'm just like dumping everything and seeing what happens.
Well, there you see how to label and color
Yeah.
your lines. So
So it's coloring things. It is okay. I can copy this. Now I will go to
why don't you put that in the chat, the dot string in the chat, and then we can all try it.
oh yeah. Yeah, this is whatever the output of the, oh, it doesn't allow because it's too big. Well, but everybody can try that with your own equations. Let me quickly just go to GraphViz online and paste that. So you can come here to GraphViz online. There's an example, beautiful example with lots of things you can do. And I'll paste this. And it draws something that looks very organized. Like this is very, very clear and organized in my opinion. And so there is a high chance it is wrong. There is a very high chance that the equations are not accurately represented because of all this process. But you have a starting point. Like in less than 2 minutes, you have a good starting point, and then you just go back and forth between diagram and equations to check if each of these arrows make sense. And then you can manually add it or ask the AI tool to make small adjustments like, oh, please convert all these arrow types to T arrows, for example. And then it does that for you. MathPix, yeah, this is the first one that shows up here. MathPix. I guess it's try the app or try for free, something like that. I have an account here. I think they have like different tiers. So there is a free tier, which I probably am. I'm definitely not paying for anything here. And yeah, possibly you can also use online, but it's very handy to just have the app here. And yeah, this is, yeah, converting to Word is one of the most powerful things here because often you are editing in Google Docs. So you can just copy and paste full the equations very nicely organized to docs from a paper.
Looks like it's free for individual users and teams. They charge if you want to use the API to embed it in your own software. So I think this looks like most of it's free for individual users.
I think we could save people a lot of trouble with that.
Yeah, that MathPix tool has been life-changing for me.
Retyping equations is a pain, and it's so easy to make mistakes. Of course, as he says, the auto-convert may make some mistakes, but you still have a lot less typing to do. Okay, so now let's come back to our friend the toggle. I know some people have gone through the toggle in some detail already, others maybe not so much. And I'd like to spend the rest of this class working on the toggle. And we started this last week, but we didn't really get very far with it, which is we have not talked so much about thinking about gene regulatory networks as Boolean networks, but we did talk about something that comes up quite often in network biology, which is what do you do if you have two signals coming in? Here A activates itself and B inhibits A. And what do you do if those signals are incompatible with each other? If the signals are coherent, then it's usually pretty obvious what to do. But if the signals are incoherent, you have to make a choice. So let's just look at this. Suppose A were 0 and B were 0. Well, then A activates A is going to provide a signal of 0 to A. B inhibits A will provide a signal of 1 to A. So is the input signal to A consistent or not? Maybe that question is so simple that it seems like a trick question. It's not a trick question. No. I have a 0 and a 1 coming into A, and I have to decide what to do about that. And if I'm doing a pure Boolean model, I basically have a choice of an OR gate, in which case I'd have a 1, or an AND gate, in which case I'd have a 0. That's really the only choice I have. If I'm in a dynamic model, then I could combine things in other ways. And I have to then think about the biology and what's reasonable. Typically, in gene regulatory networks, inhibitors are stronger than activators. And so you tend to have AND-like circuits between inhibitors and activators, and OR-like circuits between activators, but it doesn't always work like that. That's one of these things where the simplifications that we're making really have to be looked at. Biologically, you have to test. What about A being 1 and B being 0? Well, then A activates A is 1, B inhibits A is also 1. So are those signals consistent or not? Yes, those are consistent. So in that case, A definitely turns on. If A is 0 and B is 1, then A activates A is 0. B inhibits A is 0. So I have 0 and 0. Are those signals consistent? Yes. So then A is off. And if A is high and B is high, 1 and 1, A activates A is 1, B inhibits A is 0. And so in this case, the signals are inconsistent again. So I do have to make a decision about how A and B are going to combine. And it happens that the equations that I use and the equations that Herbert uses in his textbook make different decisions about how to combine those signals. And I'm going to use my approach for the rest of this class, but there are some slides which are hidden, which we're going to skip, which actually compare the results of the two decisions about how to combine circuits. So the first thing I'd like people to do is remembering how we wrote Hill functions. I'd like people to take a few minutes and write down reaction equations for this diagram. And as a hint, A and B are symmetrical to each other. So if you write the equations for A, dA by dt, then just swapping A and B, you'll get the equations for B. So you don't have to write the equations for A and B separately to begin with. If you're done before your classmates, you could try to write them the other way. In other words, if you wrote them with the AND form, then you could go back and write them with the OR form, which is how Herbert did it. And I'll walk you through it. The things we're doing in this class, in a sense, are like learning a language. And an exercise like this is a little bit like in a beginning language class where it says, translate this sentence from English into something else or from whatever language you might speak at home into English. And we need to be able to go both ways. So this is basically like that kind of exercise in a language class. In this case, we're translating from a diagram into mathematics, and I'll walk you through both directions of that, going from the diagram to the math, and then also from the math back to the diagram. And just as with learning a language, the first few times you do it, it feels confusing and you may have to look things up. And then when you do it enough times, it becomes easy. But it does take, I don't have any magic way of making it easy. It requires some practice. Although in this case, unlike with the human language, it's a mechanical thing. There is in fact a set of steps you can go through systematically to do the translation. Okay, so the first thing that we're going to do, looking at this diagram, is remembering that we're representing a gene regulatory network. And so we're going to use the gene regulatory notation of transcription factors and DNA transcription, and we're going to leave out the messenger RNA to protein step. And we have to remember also that whenever we have a gene regulatory network, we have to assume that the protein is destroyed. Otherwise, we wind up with an infinite amount of protein. And so the first thing that we do is take this basically abbreviated diagram and turn it into a little bit more complete diagram, which says A activates as a transcription factor, an activating transcription factor on promoter region of B. Sorry, A activates as an inhibiting transcription factor on the promoter region of B. B acts as an activating factor on its own promoter region. The gene B is translated into protein and then decays. So is the translation from this simplified diagram to this more complete diagram something that people feel comfortable with? Okay. So this is now purely biology so far. This is something you would see in any biology textbook or biochemistry textbook. There's no mathematics involved with it. The next thing that we do is we remember that for gene regulatory networks and regulation, we use Hill functions. And so to begin with, we're going to just look at the effect of A on A and the effect of B on B. We're not going to neglect the cross terms. And if A activates A, we know that we have a basal transcription rate B. The rate of production of A is a positive or increasing Hill function of the concentration of A. And then we have the decay of A, which is written here as a first order decay. And so now we have A activates A and B activates B. And to some extent, that's the one thing that you have to memorize, which is what the form of a Hill function is. And so whenever you see activation, you put down that template form that you see there. Now we have those cross terms. B inhibits A and A inhibits B. Again, whenever we see an arrow, we're going to write a Hill equation. But now we have inhibitory Hill functions. And remember that to go from an activating Hill function to an inhibitory Hill function, we replace the A on the top with the K from the bottom. So it's completely symmetrical. So instead of A over K1H plus A to the H, we have K1 to the H over K1H plus A to the H. And again, this is why I like the Hill function form here rather than the Tanch form. It's a little more transparent. Go ahead, yes.
So the first like inhibitory one, since A is on the bottom, that's the term for dB/dt since.
Right, it's going to be the, yes. So this term here, K to the H over K1 to the H plus A to the H, would represent A inhibiting B in this case. Although we haven't attached it to B, we've just said this is basically a label of the arrow, the inhibitory arrow, but we haven't attached it to anything yet. Now in the homework problem, it asked us to think about the strength of the inhibition. And so we're going to have a variable that describes how strongly B inhibits A and how strongly A inhibits B, and we're going to call that variable I. And we're going to say that if I equals 0, there's no inhibition. That would be equivalent to writing the two A activates A and B activates B separately with no cross arrows. If I is equal to 1, we have maximal inhibition, which would be the ordinary case, actually. As I mentioned, whenever you have what are called incoherent signals, here you have an activator and inhibitor coming in together, you have to make a decision about how to combine them by putting this I factor in, this I parameter in. We can control the relative importance of the two signals, the activator and the inhibitor. And so we're going to write the inhibition now in this form, 1 minus I plus I times k to the h over k to the h plus b to the h. Let's just remind ourselves what the Hill functions do. If I have an activating Hill function like this one, the top, if A is 0, A to the h over k to the h plus a to the h is 0. So my Hill function is 0 if A is 0. If A is very big, specifically if A is much bigger than k, then A to the h over k to the h plus a to the h is equal to 1. So this Hill function goes from 0 when A is 0 to 1 when A is large. It would be a little more convenient if it went to 1 when A was equal to 1, but it doesn't. It's a little less than 1. We also remember that for gene transcription, we have what's called the basal rate of production, B, which is that even if there is no activator, there can possibly be some small rate of production of A. And that's this lowercase b here. Let's look at an inhibitor, k to the h over k to the h plus a to the h. If A is 0, I have k1 to the h over k1 to the h, which is 1. So that inhibitory function is 1 when A is 0. If A is big, this is k to the h over a to the h, which goes to 0. And so, again, my Hill function has the normal form. If A is 0, the Hill function is 1. If A is large, the Hill function is 0. That's why it's an inhibitor. What about what I've done here? Why have I written this slightly complicated form 1 minus I? Oops. 1 minus I plus I times k1 to the h over k to the h, 1 to the h plus b to the h. Why didn't I just write I times that? Well, suppose that I is equal to 0. If I is equal to 0, I don't want there to be any inhibition at all. And therefore, I want the effect of my signal to be do nothing, which is a 1. If I want my signal to be very strong, then I want my signal to be I times k1 to the h over k1 to the h plus b to the h. Why does this have the form that I want? Well, suppose that b is equal to 0, so I have no inhibition. 1 minus I plus I is always equal to 1. So in the absence of inhibition, whatever my value of I is, I still get 1. If b is very large, then that second term is 0. And so the signal out of this is 1 minus I. So the most I can inhibit by is 1 minus I. And so that's the form that I've got there. And I've said we're going to assume an AND form. So now I take my circuit, which was A activates A and B activates B, and I have my inhibitor from B to A and A to B, and I've set an AND. An AND in logic is equivalent to multiplication, OR is equivalent to addition, AND is equivalent to multiplication. And so now all I need to do to build my full circuit is to multiply my activator functions by my inhibitor functions. And so then I have that circuit there. Herbert does things a little bit differently. He, instead of putting a product like this, takes B and puts it in the denominator of the activator function. And that has the same general effect. B is an inhibitor, but it has different effects when A and B are both non-zero. So when A is 0 and B is 1, his form and my form give the same result. When A and B have intermediate values, you get slightly different results. So now try to go backwards. Suppose that you were given this pair of equations, and I want you to take a few minutes, and we're going to translate. We've now translated that diagram into mathematics, and we're now asking you to try to do the inverse. Of course, on your notes, in your piece of paper, you've got the reverse, so you know what the result is. But try to think about it this way. Suppose that you were reading a scientific paper for the first time, and all they gave you were the equations. And suppose they were extra unhelpful and they didn't explain what the equations meant. They didn't tell you why they had the form that they had. And I unfortunately can promise you that there are many papers that do exactly that. They'll just say, here are the equations, and they'll leave it up to you to figure out what they mean. I should say that I don't approve of that, because on principle, I think you should explain why you do what you do. But people often will write the equations without thinking. And so now suppose that you were shown these equations from the beginning. Could you draw the circuit? And could you explain what each parameter in the circuit means? And I'm asking you to do this because if you're going to do a project, you're going to have to be able to make those translations. If you're going to take a paper that's a published paper and you're going to be able to understand it well enough to present it as a project, you'll have to be able to go back and forth between the mathematics and the underlying networks. And so I want to practice this together here in class. And then you can practice it on your own. You could find diagrams and try practicing it on your own to begin to build up the ability to translate back and forth between these representations.
Like how Herbert draws it differently. Like I get it when it's like this form because it's very, each term associates to something, but is there something more generic of like, if there's another, I don't know, terms on the top will be promoting, and terms on the bottom will be like inhibitory kind of.
Right. Right. So the comment was, what if you don't have this product form, but you actually put everything into one big hill function? And when you're doing these combinations of signals, there are a number of different ways that you can do that combination. You could write a to the h plus b to the h over k1 to the h plus a to the h plus b to the h. That would be one way of doing activation. You could write you could write a plus b quantity to the h over k1 to the h plus a plus b quantity to the h. That would also be activating. Or you could write a to the h over k1 to the h plus a to the h times b to the h over k1 to the h plus b to the h, which would also be activating. All three of those are going to have the same behavior, that when a and b are both big, it's on. When a and b are both small, it's off. But for intermediate or conflicting values of a and b, they're going to give you different results. Your comment, which was a useful one, is, again, and this is how we learn language, we look for patterns that we can recognize. And you made the comment that if you see the variable in the numerator of the Hill function, it's functioning in an activating way. If you see the variable in the denominator and not in the numerator, it's functioning in an inhibitory way. And so in this case, in the one that I've written here, we have a to the h in the numerator, so it's an activator. We don't have b to the h in the numerator, so it's an inhibitor. If I had Herbert's form, which would be a to the h over k to the h plus a to the h plus b to the h, I still have a to the h in the numerator, so it's an activator. I have b to the h only in the denominator, so it's an inhibitor. So both of those have the same relationship where a is activating and b is inhibiting. So that's a great way of looking at these and trying to analyze that. In principle, for some of the metabolic networks, for example, that one might look at, where you might have a lot of components coming in, a lot of enzymes at the same time, you might have a lot of things that are coming in at the same time. So you might have a to the h in the numerator, b to the h in the numerator, c to the h in the numerator, d to the h in the numerator. And then in the denominator, you'd have all of those plus some inhibitory factors as well. So, but the point here is that you don't have to panic, no matter how complicated the system looks to begin with. Looking to begin with, as you said, look at the items in the numerator. Those are going to be activators. Look at the ones in the denominator. Those are going to be inhibitors. The exact way that they're written out will have effects when they're mixed states. But you can still say if they're all ones, what happens? If they're all zeros, what happens? That was a great insight. So let's just walk through our diagram now. And now let's go back from the mathematics. So let's walk through this. We're going to start from the simplest thing. Remember that for a gene regulatory network, even if we don't have any activator, we may get some basal transcription rate. And that basal production rate is this K times B. And that corresponds to this hooked arrow here, which is the production of A independent of what my input signals are. And so K times B, the bigger B is, the more I get even in the absence of signal. So that's the basal production. The next thing is the self-activation. This is A promotes A, B promotes B. That's my Hill function, a to the h over k1 to the h plus a to the h. And of course, it's also multiplied by k. And that would represent this activating arrow where A activates itself and B activates itself. Does that make sense so far? Now we go to the next step. What's the maximum velocity of production? The maximum velocity of production is K, V max. That shows up here. That's determining the maximum rate at which that protein is translated. As we've said before, whenever we have a gene regulatory network, we have to infer that the chemical decays, A goes to nothing, and we're using what's called first order kinetic. The rate of decay is proportional to the amount we have. That's this arrow here, and that has a rate K2. And it's worth pointing out that the equilibrium concentration of A will be approximately K divided by K2. That won't be exact, but it'll be approximately that. The bigger K2 is, the faster A decays, the less A I'm going to have in my system. The bigger the production K is, the more A I'm going to get per unit time. And when those two things balance, I'm going to have something that's proportional to K, little k over K2. And if we'd done more work with steady states, we would have spent a lot of time analyzing. And if that's not familiar to you, there's a chapter in Herbert's textbook, and also that math textbook that I suggested to you goes into a lot of detail on that analysis. Okay. And now I have the cross inhibition, which is that inhibitory arrow, that T arrow, and that's my K to the H over K1 to the H plus B to the H as that inhibitory form. And I have to make an assumption about how my activator and inhibitor interact. And in this case, I'm multiplying the two things together. So that's what this is doing here. And finally, I have the strength of the inhibition, which represents these arrows here. And that's the 1 minus I plus I here. And so each term in the equation corresponds to one of the components of the arrows in my diagram. Each parameter in my equation corresponds to some fundamental biological concept: the basal rate of production, the self-activation maximum velocity, the strength of inhibition. The one thing that we didn't talk about was that capital K, the Hill constant, which is the concentration at which the rate of production is half maximal. And in this kind of equation, we're scaling everything between 0 and 1, so that capital K is going to be equal to a half. As I mentioned in Herbert's textbook, he likes to scale everything between 0 and 20, so he sets K equal to 10. Hold on. Go ahead. Yes.
For the I, like strength of inhibition thing.
Yeah.
Do we determine if it's 0 or 1 based on like if B is high, it's 1, B is low, it's like it's dependent on B, or?
So the question is, does the value of I depend on B? And the answer is no. B is a variable in my system. B is the amount of protein of type B. I is a fundamental property of the network. It represents how tightly coupled the circuit is. In other words, suppose that I am trying to talk to people in this room, which is quite noisy. I would represent essentially how much sound is getting from me to you. I'm having to talk very loud so that you can hear me over the air conditioning system here. So if I is equal to 1, then the signal from B to A is getting through very loud. It's getting through 100%. if I is equal to 0, the signal from B to A is getting through at 0%. The signal isn't being received. Now, there could be things in the environment of the cell that modulate how strongly B affects A. There could be a separate molecule C that controls the strength of inhibition. In that case, what you would see would be there would be an arrow pointing to the arrow between B and A. And in that case, I would be a function of C of that concentration. But in this simple model here, I is something that we're given. And we can ask the question, given the value of I, how does the system behave? We know that if I were equal to 0, we have A and B totally decoupled. A activates A, B activates B. That's all it does. And we already studied a few weeks ago what that circuit does. That was that bistable circuit. And so the idea here is that if we start with I equal to 0, A and B are completely independent. And now we gradually increase the strength of I from 0 to 1. We're gradually making the coupling between them stronger and stronger. When I is equal to 0, these are independent circuits. Now we're going to say what happens as we make that coupling strength bigger. And that's actually very much the kind of experiment one would do in, say, a metabolic network, where you say, what if I change the amount of an enzyme? What happens to the circuit? So I might represent here we don't have it explicitly represented, but if I depended on some other factor, then we could be changing the strength of that coupling.
So it's not always 0 or 1.
No, I could be any value in this case. You could legitimately ask me, what kind of biological situation might I not be 0 or 1? And I could come up with a variety of situations in which it isn't. But in this case, we're treating this as a fairly abstract network. I'm not saying this is particularly SOX and OCT, for example, where I know the specific molecules. If I were talking about a specific molecular toggle, and there are many of them, then I would have to begin to say, what is it specifically that modulates the strength of that inhibition?
So something like this just allows you to like simulate the dynamics and say, okay, with no inhibition, it goes like this, and then you
Right.
put it on, and then you.
That's right. Right. That's why these have the name motifs. These are patterns that have typical behaviors. And we will see those patterns played out in more or less the same way in real biological circuits. But they may not be exactly the same. The real biological circuit will be similar to, but not identical to these patterns. On the other hand, the patterns have very strong types of behavior. And so, for example, Herbert's version of this circuit and my version of the circuit have very similar behaviors. They're not exactly the same, but they're pretty much similar. Yeah.
So I activation is right. So I understand Henschel's the Hill coefficient. But like in the about the K, so it said like it's an inflection point. So what exact it does like what
So remember that's what we started with here. Let me come back to the beginning here. So the question was, what does the K, capital K, the Hill K do? And that's why I wanted to remind people here. So the structure of this function is, if the concentration A or B is bigger than K, capital K, then the output of an activating Hill function is nearly 1. If the value of the concentration of A is below K, the value is nearly 0. Near K, the output is about 1/2.
So how do we like actually determine K for all these reactions? Like, does it will vary with respect to conjugations?
So the question was, how do we figure out what the value of K is? And so, again, if we were doing more mathematical biology, a fundamental concept, and I'm regretting a little bit skipping some slides about Hill functions at this point. I was trying to avoid too much math, and maybe I bought myself a problem by doing that. Is a fundamental concept in mathematical biology is you do what is called de-dimensionalization. So, for example, when you're talking about time, you don't measure time in minutes or seconds. You might measure things in units of what was the cell cycle time if you're doing cell reproduction. Or you might measure things in terms of what is the typical time it takes for a molecule to get from one place to another. So you measure things in terms of natural units. And if you want to do molecular circuits, you want the values of your variables to be roughly the same. And so if you have one molecule that's active at a millimolar concentration and another molecule that's active at a nanomolar concentration, it's not very convenient to try to add millimolars to nanomolars, because when you do that, basically the nanomolar disappears. However, some things are equally, what matters is the level of significance relative to that K max. And so what we've done here is essentially say, whatever that K is in real life, we divide the concentration by that and measure in units of that. So, for example, if we were dealing with glucose, where the real concentrations are quite large, or in millimolar, then the K, the actual K in units, would be in something in millimolars. If we're dealing with transcription factors in the cell, where transcription factors might exist in 1 or 2 copies inside the cell, maybe a few copies inside of the cell, those might be in nanomolar concentrations, and they might be equally active in nanomolar concentrations. So if we want to convert from these abstract mathematics back to an experiment, K is whatever concentration in millimolars, nanomolars, moles, the chemical has half of its maximum activity. And the nice thing is that if you divide the concentration by K, that's dimensionless, because K and P have the same units. They're both concentrations. And so once you have a dimensionless form, you can combine things in a way that's consistent. The moment you put things back into molar forms, you can't combine things freely anymore. And so you're absolutely right that I did not go over the fundamental concept of how to do de-dimensionalization or scaling. That's an important concept. The textbooks, Herbert's textbook talks about it a little bit. The textbook that I recommended, that I sent around in the email, discusses it in a lot of detail. A standard mathematical biology textbook in chapter 1 or 2 will talk about how to do that. And so, for example, that Essential Mathematical Biology by Britton will go over this in detail. And if you wanted to, I'm sure that if you Google the question of de-dimensionalization, you'll find a pretty good description. I don't remember if Wikipedia has a good description of this, but it would surprise me a bit if they don't. They tend to do pretty well with that kind of concept. But the basic thing is that you want to be sure that you're not having to connect the numerical values of the concentration. What you really care about is the level of activity relative to this reference K. Now, there was a separate question, which was, can I measure capital K? And the answer in general is yes. If I do an experiment where I change the level of P, I can measure the rate of production of the chemical or the equilibrium amount of the chemical. And I can ask the question, what is the concentration of P at which I get half of my maximum rate of production? And so that's actually, that capital K is something that can be measured at equilibrium. It's not a kinetic constant. And therefore, that's actually something that's relatively easy to measure in experiment. And so you'll see for lots and lots of chemicals, you'll see that capital K, that half maximum concentration. If you get a sigma chemical catalog, it'll tell you what the half maximum concentrations are for enzymes.
In solution or something, they run those experiments?
Well, so there's a good question, which is, under what conditions is that K measured? And the answer is that depends. And so it's certainly possible that under different conditions that K will be different. As you change the temperature, that K will be different. As you change the pH, the K will be different. As you change the salt concentration, that K will be different. And so there will be some reference set of experiments which will tell you how that was measured. For our purpose, we're imagining that the cell is a working cell. And so we're in the cytoplasm of the cell, and it's whatever the environment of that particular molecule is in its normal state would be where that K is measured.
Do we know, and is it pretty consistent to like pH inside of the cell?
That's a good question. Cells definitely regulate their pH.
It is very stable.
In like temperature and all those things we expected, or is that?
Temperature depend on the tissue, like skin for sure, like internal organs, it is very well maintained, but not because of the cell itself. The cell itself doesn't maintain temperature, but it does not maintain pH very, very well.
So I'm probably not knowledgeable enough to answer that question. There are tissues in which the pH is different. There are cell types in which the pH is different. It's also not always completely obvious that pH by itself is going to be the most informative thing for a cell, because the pH will depend on the concentration of a lot of ionic species. So the cell will regulate calcium, potassium, sodium separately. And so the pH will depend on those ionic levels for all of those species in some relatively complex way. There'll also be hydroxyl and other ionic species there.
Yeah.
And even within the cell, there'll be things like lysosomes or peroxisomes, where inside of that vesicle, the pH is strongly different from the pH elsewhere.
Yeah, mitochondria too.
Mitochondria would be another example of that. And for many viruses, the unpacking of the virus, the viral genome depends on being taken into a vesicle where the pH is different from the environment outside the cell. And also the repacking of viruses into virions is often triggered by pH changes within the cell, which indicate that the cell is becoming energetically damaged. So the lysis of the cell and the viral packing will be triggered by a failure to maintain pH inside the cell. So that's a great question. It's also one that reference database, Biology by the Numbers. I haven't actually searched it for pH, but if you asked it, what's the equilibrium pH inside a normal cell of a given type, it might give you some information about that. And of course, there are all sorts of specialized cells. I mean, you have microbes, you have things like archaea, which can live at very high pH, very low pH, very high temperatures, very low temperatures. And so there are all sorts of single-celled organisms where all of the cellular enzymes are different because they're adapted to work at different pH levels.
Yeah, but it is well controlled most of the time. If it's not controlled, probably the cell is under stress.
Yes.
Because pH changes structure of protein and structure of DNA. So if pH changes too much, the cell is probably gonna die.
Again, for our purposes, when we build a model like this of gene regulation, we are making so many very harsh assumptions that I think that assuming that the pH of the cell is relatively constant is probably the least of our worries.
Yeah.
Which is not to say it's not a good thing to ask. I think it's a very important question to ask. And as I mentioned the other day when I said that I had been reading some Wikipedia and it took me to this last common ancestor question. The reason I was looking at that was to read about, current thinking about epigenetic regulation of transcription and translation, all of which are totally ignored in a diagram like this. And I have to say that an awful lot has been discovered about those since I studied biology, which is quite a while, which is not that long ago, but long enough ago. 30 years ago, many of the mechanisms of epigenetic regulation were not known. Right. RNA interference was only discovered about 15 years ago.
I think I learned that 98% of the genome was jumped in.
Right. It means we didn't know what it did. But now all those RNAs that people thought didn't do anything actually wind up doing a lot. Almost none of those are included in these kinds of diagrams. And as I mentioned, in eukaryotes, the transcription of genes is heavily influenced by methylation of DNA, acetylation, methylation of histones, acetylation of histones, and all of those things are not included in this kind of model. And I find the biology fascinating. But we also, at some level, are stuck with the simple mathematics. First, we have to do the simple mathematics, and before. Our hope is that if we can do the simple mathematics, then we can gradually add complexity on top of it. But these are all great questions to ask. So now we come back to, here's their circuit. And I'd like you to implement this circuit in Antimony and run the time series. And I want you to do a parameter scan where you change the initial values of A. And I'd like you to plot on two separate plots, A versus time and B versus time. You should have some version of that code already running for you, in which case doing this shouldn't take too long. You may have to change the molecular circuit a little bit, but it shouldn't take too long. And if some people get this done faster than others, you can try changing the values of I and K2 and lowercase b and see how things change. As I mentioned before, almost everything we do in this class is progressive, and so it's important to keep the notebooks from one class because you're going to always reuse them in the next class. But this is an exercise in first plotting a time series and then doing a parameter scan and putting the values from the parameter scan on top of each other.
And when when A is equal to B, basically they have to stay together all the way. They have to go like straight like this without going down. But because of numerical instability, eventually it collapses to one of them because the solver freaks out.
You can also update the solver settings to make them more accurate. So one thing you should try to develop some intuition about is what time do you have to simulate for? In this particular set of parameters, everything happens within a time of about 10. Well, you can use any value of initial value B you like. You'll get a slightly different plot. It's worth exploring that. Now change the initial value of B0 and try again, see what changes. Do the equilibrium values of A change when you change the initial value of B? Does the final value of A change? When B is 0, A should always go to 1, yes. But try intermediate values of B. I could give away the answer. If initial value of A is bigger than the initial value of B, A goes to the high value. If the initial value of B is bigger than the initial value of A goes to the low value. If A equals B to start with, they stay equal unless there's some problem with the solve. And this is actually almost exactly a repeat of the exercises we did in the third lecture for just A activates A. All of the code that we're running, all of the loops that we're doing are things that we already did 2 weeks ago. So it's important to save that code and remember how to reuse it because you don't want to have to reinvent it every time. Depending on your initial value B, you're going to get different results. But you'll notice that the fixed points don't change. Whether you go to A high and B low or A low and B high will depend on the initial values, but the final values will be independent of that. Now go back to your values from before and change that little b, the basal transcription rate. We set the initial value of B equal to 0.5. Try 0.1 and try little b equal to 1. Is it different? If you make little b, what if you make little b 0? What happens? You should see something interesting as you change the value of little b. Somebody tell me what happens when that basal transcription rate is 0. What happens? Both of them go to 0? A and B both go to 0? And what if you make lowercase b equal to 1? What happens? So you should have a program that generates a plot like the one on the bottom right. And now what you want to do is change the value of little b, make it 0, make it 0.1, 0.2, 0.5, 0.7, 0.9, 1, and see what happens to that plot on the lower right. What happens? And this is a very important part of understanding how a system works. this is called sensitivity analysis. You go through and you systematically change each parameter one at a time and see what the effect of that single parameter is on the system. So what happened when you made little b equal to 1? Did they depend on the initial value anymore? I want people to be generating this plot on the lower right. That's what I asked you to do in the previous exercise, and now you're going to change the value of little b. You do that by hand. What happens? So why don't you try repeating these experiments? Lowercase b equals 0, 0.1, and 0.18. And these are the values of A for different initial values of A. See if you can repeat these. And you need to plot both A and B time series, but on separate plots. To get the basic result takes a minute. There are all sorts of things that you could do in terms of adjusting how long you run it for, what the resolution in terms of the number of the range of initial A and B is, what the spacing is between them. All of those things can take a long time to make pretty. So you'll notice, for example, here in my x-axis, that the time scales are different for these 3 runs. Why are the time scales different for the 3 runs? Because I noticed that when I went to b equals 0.18, at time 10, which I used initially, things hadn't equilibrated yet. And so I noticed that, and then I went to a longer time run. So that's the kind of thing that you have to begin to develop an intuition for, is you look at your time series output and then you adjust your settings for the time series based on what makes sense. Another thing that you'll notice is that even though at long times, both A and B go to a low value, you see there's this sort of elbow or kink where the value of A gets hung up around 0.5 for a long time. And if we were doing dynamical systems theory, I would be able to explain in some detail why that is. And I notice in this graph, the two plots on the far right, the first digit, this is not 0246802. The 1 point got cut off because of the overlap of the images. If you plot A and B in one graph, it's very hard to distinguish them.
Yeah.
It gets confusing.
No, I'm trying to understand the oval thing.
That's the unstable fixed point. When you get close to an unstable fixed point, your time evolution slows down. For small b, you always go to a low final state. A and B both turn off. They both turn off, regardless of the initial value. Now, here is high values of b. Try b equals 1.3, 1.5, and 2. Try that. In that case, no matter what your initial value B and A are, they both go high. Again, you're only changing one number in your plot. Possibly 2, because you might change the duration of the simulation. And now in the middle, which is where we started, for B equals 0.3, 0.8, depending on whether A is higher than B or lower than B, you get a split to high and low values. So now I can summarize this. If the basal transcription rate is low, no matter what my initial condition is, both A and B turn off. If the basal transcription rate is too high, no matter what my initial value of A and B are, they both turn on. For intermediate rates of basal transcription, whichever starts out higher, A or B wins, and I'll wind up with high A and low B, or high B and low A. That is, the latch only works for an intermediate level of basal transcription. So now I'd like you to write a simple function, and we already did this 2 weeks ago. So you can use the notebook that you had then, where you scan B between 0 and 2, calculate the steady state values of A, and you'll have to do this for A starting low and A starting high, and plot the steady state values of A versus lowercase b. So you're basically taking the plots that you just did, taking the long time value of A. In some places, there'll be one long time value. In some places, there'll be two. And you're plotting that against the value of B. We call that a bifurcation plot. And we did that 2 weeks ago. And you have the code.
It's unclear to me how, even like for low B, like in my imagination, even for B equals 0, if you have high A, low B, you have a positive term there. In the end, you have basically K times A over K to the n plus A. The other one is 1, because you have B equals 0, and then minus K to A. So you have a steady state of given by the ratio of these terms, which is non-zero.
That's a reasonable question, which is, why do you need to have a non-zero basal transcription rate for this circuit to work? And that comes about because of the AND that we've applied, the multiplication of the two terms. And that's the reason Herbert uses a different form for the cross inhibition. In Herbert's Hill function, you don't need to have a basal transcription rate. Basically, because you have A and not B here, the AND function has a tendency to turn everything off. And so you have to have some leakage to get anything to happen.
Yeah, but if it's 0, hard 0, the second term is 1. Yes, if B is 0, the second term is 1. And if A is 1, the first term is a constant. And then A goes to some positive value, depending on K and K2. The only way would be if A is bringing B up. But if if small b is 0 and uppercase B is 0, dB by dt is always 0.
Well, try it. See what you get. I mean, A and B don't go to 0 exactly. They go to a small value, but not to 0. If we made the decay rate smaller, remember the decay rate K2 is quite large here. If we made K2 smaller, it would be different. We've got K2 equal to 1.
But even in that situation, if uppercase B is 0, starts at 0, the first term in the PDT is 0.
Right.
Everything is 0. Everything is 0 if B is 0. Lowercase and uppercase B are both are 0. Everything is 0 in the BDT. Which means A should go to a positive non-zero steady state.
So if you start with B equal to lowercase b equal to 0, capital A initially to 0, then the first term is 0, and so dA dt is 0, so you stay there.
Yeah.
And that's an unstable fixed point. And we're specifically avoiding that. We're not looking
Yeah.
at that case.
So anything that is not hard 0 will move B up and then will
Right.
bring A down, and then both will converge.
Right.
But that's very, very non-trivial to spot.
Well, that's why you need the phase diagram.
To get
But to get both branches of the high one and the low one, you have to start with A bigger than B once and B bigger than A once. As long as you have both cases, you'll get both branches. So if you got that to work, you could try doing the same code, but instead of scanning B, try to scan I. The interesting range for B will be slightly different as I changes, but if you pick B equal to something like 0.7, that's a good choice. You just made a very fundamental point, which is that the parameters actually do interact with each other in interesting ways. So scanning just one at a time isn't quite enough, but it's a good place to start.
Can I only be between 0 and 1?
Well, you do, if you make I bigger than 1, then you can get a negative value, which you don't want. Now, to get the unstable fixed point, there's a bit of a cheat here. I had suggested something in the slide which doesn't work. If you start out with A equal to B, and then you don't run it very long, you'll get an intermediate value, which is the one that you were seeing before, where it was, it was staying there and then jumping away. So for this plot,
For the B's?
if you start out with A equal to B, capital A equal to capital B, you'll get a line that goes in the middle. Try that. So you're now going to plot 3 lines instead of 2. You'll have to run it again with the initial value of A equal to the initial value of B. And if you run it for too long, you'll get, it will jump off to one of the high or low values. That was what you were seeing earlier. That jumping that you're seeing is a numerical instability. It should stay in the middle, but the integrators are,
And then you just get like a.
that's perfect. So now try to overlay that on the other ones. So the next, you got the unstable fixed points too. So did you get something that looked more like that?
Oh,
So you'll not, so while we were looking at the value of B, as we change the value of B, you'll notice that the steady state of A goes from having one value when B is small to 2 possible values for intermediate values of B, and then 1 value again when B is big. And that switching from 1 possible value to 2 values and then back to 1, those are called bifurcations. When the number or stability of the fixed points changes, that's called a bifurcation. You've plotted the upper branch and the lower branch. There is an intermediate branch, which you've already seen, which is start with A and B equal to each other. A0 equal to B0. Starting with a value of 1/2 is not bad. A equals 0, B equals 0, 1/2. And don't run it for very long because you'll get numerical instability. So run it for, say, 10 time steps and plot the final value, and you'll get a value that's intermediate between the high value. For low values of B, they'll be the same. For high values of B, it'll be the same. In the middle, it'll go in the middle. Try that. And you may want to go back and re-examine those exercises that we did for that homework 3, because these are all the things that were asked for in homework 3. And so if you got it done, wonderful. If you didn't get through all of it, now you have more of the tools to do that. And so now that you have this code at home, you can try scanning. Instead of changing the B parameter, you could change the I parameter.
Oh, no.
Or you could change the K2 parameter. So when you get home, try scanning I instead of B, and you'll get something that looks like that. Or if the inhibition is weak, you get high values, and the same thing for the unstable branch. And so you can then gradually go and you can explore the behaviors in each one of these parameters. And this would correspond to an experiment where it's not a complete knock-in or knockout, but where you have some regulation, where you partially inhibit a pathway or you partially activate a pathway. And in this case, what you see is that for some low value of the enzyme, you get a single value. For a high value of the enzyme, you get a switching behavior. And that's a very fundamental thing that happens. And this circuit is particularly important in development when you have stem cells that are differentiating. A circuit like this controls stem cell differentiation. And there is, in fact, precisely something where the level goes up so that the cross inhibition initially does not act. And then at a particular time, the level goes above some threshold and the cross inhibition locks you into either one state or the other.
Depending on your value, your low or high value.
Whether you were initially high or low, yes, that's right. That's right. So in the early embryo, there's a selection of cells that are going to form the embryo proper and then the extraembryonic tissues. And that's controlled by a latch of this kind called the Sox-Oct latch. And there's a bias term that pushes you in one direction or the other. That bias builds up and then forces the cells. Initially, the cells don't choose either state. They could stay in the middle, and then they're forced to choose one state or the other. And so actually what we're seeing here is something that really happens in development. This is a pretty abstract way of representing it, but nevertheless, if there weren't this bifurcation, we'd all be stem cells. There wouldn't have ever been a choice between stem cells and non-stem cells. If you want to explore, there is a notebook that I've created, which is in the student materials folder, which is called Null Lines and Toggle. And it has code blocks that generate not only the bifurcation diagram here, but also these phase and flow portraits. And I talked a little bit last week about what these mean, but I'll talk more about that next week. Those really will help you understand. Pedro was asking a question about why you get the pattern of behavior of high and low values that we were seeing. And these flow portraits that we're going to look at next week a little bit help you understand that. We're not going to go into great detail about those. And the reason for that is they're really useful if you have two variables like this one where you have A and B. If you have three variables, you can plot them in 3D. But if you have a lot of variables, you have 8 chemical species, the same bifurcations and things happen, but you can't visualize them. And so traditionally, we would spend a couple of lectures on those, on bifurcation diagrams. But if we're going to be doing more complex circuits, it's important to understand the basic idea of a bifurcation, but actually all the tools to visualize it don't work when you go to more complex circuits. But if you look at Herbert's chapter 13, the whole chapter is about that.